Hello, my name is Allison Gilchrist. I am a research scientist at NYU Langone Health, where I study vaccinology and immunology. Today, I had the pleasure of interviewing Dr. Barney Graham, who is the keynote speaker at our upcoming Keystone Symposium on progress in vaccines development for infectious disease. Well, it's lovely to meet you, and thank you for agreeing to this. I was hoping we could start with a couple of the more personal questions. So the first question is, if you could tell me about your career trajectory and what attracted you to the government sector.
My career has had a lot of twists and turns. I started out going to medical school planning to be a small town doctor, and after residency training I decided to do an ID fellowship. I did some chief residencies, and then I got a PhD, and I progressively tried to engage more and more in basic research, but kept clinical research as an important part of it as well. And after 21 years of Vanderbilt, that included a lot of that training, I had an opportunity to go to the Vaccine Research Center, which was a new entity that was being built on a NIH campus in Bethesda. And the mission of the VRC was to develop an HIV vaccine. And my role primarily was to develop and build out the new vaccine evaluation clinic. And that's what my original initial focus was, but over time, because I also had a basic research lab that mostly focused on respiratory syncytial virus, I was able to continue a lot of my work on viral pathogenesis and vaccine development. So eventually, in 2012 or 13, Dr. Ledgerwood, one of my fellows at NIH, took over the clinic, and over these last 8 years or so, I've been able to focus primarily on laboratory work.
Great. And how did you become interested specifically in structure-based vaccine design? So what kind of drove the interest of your lab in RSV and then in later work as well?
Almost all of my work in science has been done somewhat opportunistically by noticing technologies that were emerging and other fields that I could apply to respiratory syncytial virus pathogenesis work. And at the Vaccine Research Center, a major component of that effort to make an HIV vaccine was structure-based design and structural information on the envelope glycoprotein of HIV. That effort was led by Peter Kwong at the VRC, and in 2008, Peter got a new fellow in the lab who had done structural biology training at Johns Hopkins, Jason McClellan, and Peter's lab had gotten so big on the 4th floor that Jason had to have a seat on the 2nd floor where my lab was. And Jason wanted to do more than just HIV, and I encouraged him and persuaded him to work on respiratory syncytial virus, which didn't really have much of any structural information yet. And so we focused on the F glycoprotein, and we solved the structure of a couple of the epitopes, and then the structure of the post-fusion form of the RSV-F protein, which is its spent or non-functional, its post-fusion form. But the real goal was to find what it looked like in the beginning, how the protein looks to begin with, and that is the pre-fusion form. And eventually we were able to capture that. Peter's lab and my lab, with Jason doing a lot of the hands-on work, ended up finding the structure of the pre-fusion F protein by having an antibody holding the top together and using the trimerization domain to hold the C-terminus together. And having that structure, having that image, turned out to reveal some very neutralization sensitive parts of the protein. And That then led to efforts to stabilize the protein to keep it in that shape because it's otherwise very semi-stable. It's metastable. It doesn't hold its shape very well by itself. But once that prefusion form is constrained, and you can preserve those epitopes at the apex, it becomes a very good vaccine antigen, much, much better than what had been used before for RSV. So solving that structure all of a sudden gave you a pathway toward having a better RSV vaccine. And that work on structure really changed the field of RSV. And as Jason moved on to his faculty position at Dartmouth and now later on to UT Austin, we wanted to extend that understanding of structure on other envelope viruses, especially those that used class 1 fusion proteins. And so we started a collaboration on coronaviruses because there wasn't any structures known yet for coronaviruses at that time. And so working with Jason and then later with Andrew Ward, trying crystallography, but then having to apply cryo-electron microscopy to find the structure, we were finally able to get a structure of the spike protein of a human beta coronavirus. It was the HKU1 endemic form, endemic coronavirus. And again, finding ways to stabilize that protein in its prefusion form, not only preserve those neutralization epitopes at the apex of the molecule, but also made it express much better. So it gave it an advantage for gene-based vectors. So, and now we've become so spoiled. Once you understand and can see the atomic level structure of a protein and know how to define precisely the epitope targets of the antigenic binding sites that are needed to actually make an immune response effective, especially for antibodies, once you have that, you don't want to go back. You always want to have structure. And so for me, it would be very hard to start any kind of new vaccine development program without knowing the structure because it's now feasible because of all the advances that have been made by Peter's group and others, mostly because of efforts to make an HIV vaccine.
That actually segues really well into a more scientific question, which is how did previous research on coronaviruses and mRNA vaccines, you can focus on coronavirus structure if you wish, specifically prepare us for the SARS-CoV-2 vaccine development program? What was your expectation of timeline to deploying the vaccine, and were you surprised by how quickly this happened?
Right, well, it was not easy to get the first coronavirus spike structure. It took more than 2 years because trying it with the MERS and the original SARS-1 coronavirus spikes it didn't work out. They were too unstable. We could never get a uniform structure. It wasn't until we turned to the endemic coronavirus HKU1 that we were able to finally get the structure. But once we had ways of stabilizing that, especially the 2 proline substitution at the top of the central helix, that same exact substitution worked to stabilize the MERS and the original SARS and about a dozen other beta coronaviruses. And so we had a generalizable approach that seemed to be working across the other members of the coronavirus family in hand that was part of our pandemic preparedness program. And so that gave us an opportunity to rapidly solve the structure of the new coronavirus once we had the sequencing and order some of the reagents, and that was done in Jason McClellan's lab, and it was allowed us to launch the vaccine program, because once you can see that the protein, the antigen you want for the vaccine is in the right shape, has all the right surface contours, it really gives you a lot of confidence on going forward. Now, the RNA is a different story because we've been working with DNA vaccines for 30 years, way back into the 90s. And a big part of the VRC program for HIV and other viral diseases was to use DNA vaccines. And they had worked reasonably successfully, especially for flaviviruses like the West Nile and then Zika. But in 2016, during the Zika outbreak, when we made a DNA vaccine that was reasonably good and went all the way into phase 2B trials, we also worked with other groups, including Moderna, who had made an RNA vaccine that was designed in a very similar way to our DNA vaccine, but it was much more potent. And it was also even faster, even more rapid to make RNA vaccines than it was to make DNA vaccines. And so that capacity for chemical synthesis and rapid manufacturing combined with our thinking that we had a generalizable approach for vaccine development, it was a strategic plan with Moderna to do pandemic preparedness demonstration project for both paramyxoviruses, where we also had another approach, and coronaviruses. So we had been doing that with Moderna for about 3 years before this pandemic started. And so for both the antigen design component and for the nucleic acid delivery component, this had involved a lot of work over many, many years, and then they all came together right at the right time at the beginning of 2020.
One of the questions that I had planned to ask was, did you expect a global pandemic of this proportion during your tenure at NIAID? And it sounds like yes, or at least you were prepared for it.
Well, we felt like our job at the VRC was to get ready. We didn't expect it, we didn't want it, but because of these new tools that had emerged over the last 10 to 15 years with structure-based vaccine design, having atomic level structure, knowing how to do protein engineering to hold the proteins in the right shape. There's new technologies now for displaying antigens on self-assembling nanoparticles. There's the capacity to do single cell analysis and that can be helpful for discovering new human monoclonal antibodies, but also for analyzing immune responses in much more detail, rapid sequencing. There's a number of new things that have happened over these last 10 or 15 years that really were transforming the way you could do vaccinology. And because of that, we wanted to find ways that could have generalizable use in case we did have future pandemics. And everyone thought, including me, that the next big pandemic threat would be influenza. And so our influenza program was also very active, and we were working as hard on influenza as we were on these generalizable approaches for other viral families. But the NIAID plan is really to have look for generalizable approaches for interventions and medical countermeasures for all 26 viral families that infect humans. So we had started with paramyxoviruses and coronaviruses because we knew the most about them, but we really think this kind of work needs to be done for all the viral families.
Mm-hmm. So you had a very unique perspective as the deputy director of the Vaccine Research Center, and you talked a little bit about preparing for a pandemic. After the preparations that you had in place, did the pandemic proceed as you expected, or as you had prepared for? What do you wish had gone differently in the response, on the response side?
Yes. Well, near the end of 2019, we actually had a plan with Moderna to do our demonstration project with the paramyxovirus construct that we had made for Nipah virus. And at the end of 2019, we had designs for Nipah virus and a generalizable approach for paramyxovirus and for coronavirus. And we had animal model data showing that each of those could be delivered by mRNA and protect against lethal challenge in animals. So we were planning to do a phase 1 trial with the Nipah F protein. It's actually FG chimera protein for just a demonstration project in a phase 1 clinical trial using the mRNA delivery. But after the 6th or so of January, 7th of January, when we found out it was probably a beta coronavirus, we flipped our demonstration project to coronaviruses and for this new viral sequence once it was available. And so initially, this started before we had our first case in the United States. Our initial goal was to do a demonstration project to show what was feasible, to show what you could do in case you needed it. And so our plan originally, initially was to go as fast as we could, and whether it was a pandemic or not, it was a demonstration project. And so we did that, and it turned out that it turned into the real thing because, as over those next 8 to 12 weeks, after those next 8 to 12 weeks, the pandemic did emerge.
Yeah, and what you had planned for the demonstration project, I guess, did everything proceed as you had hoped it would in terms of ramping up and getting governments and scientific agencies on board?
For the demonstration project, initially it was just Moderna, and at that time Moderna was a relatively small biotech company who had a good technology, but had never taken a product into licensure or into the market. And so our plan was, and what we thought could make this fast, is that we would design the vaccine antigen and manufacture it and take it into a clinical trial without any additional experimentation. And at the same time, in parallel, we could look at a variety of other constructs and see if any of them were better than what we had started. But the idea here is to have a generalizable approach that can be immediately applied and immediately go into manufacturing and then immediately go into the clinic. And so those things that we had planned that were especially good for coronavirus, it wouldn't have gone nearly this easily if it had been something other than the coronavirus, because that's the one we had the best generalizable plan for. And so the first sequence, the first design we selected turned out to be as good or better than any of the other ones that we had tested. And so we got off to a quick manufacturing start, and we're able to get the phase 1 trial started within about 65 days. If we had had even more preparation, and if we really knew this was the real deal, I think it could have probably started in about 40 days if everything was all lined up. And if you look at the entire development process and everything that has to be done to get through the regulatory process and into phase 3 and a result, I think if we had had all those things lined up and a plan ahead of time, you could have even gotten this done within about 8 to 9 months instead of 10 or 11 months. So I think we could have gone even a little faster if we had been even more prepared, as I think we should be in the future.
Yeah, can you expand on that? Like, what have you learned from this pandemic and the response to it, and how we can apply that to future pandemics and be more ready in the future?
I think one thing we've learned during this pandemic is that talking about a problem or talking about a future catastrophe is not the same as responding to a catastrophe. And And that really, as much as we had discussed and talked and planned, we were not really ready for a pandemic. I don't think any place in the world was really, truly ready for a pandemic. And everything from the regulatory process to the deployment process, once you have a vaccine, how do you get it into the arms of everyone in the world so you can really stop the virus before it is spreading as far as it has. All the steps from beginning to end required a lot more work and a lot more on-the-fly organization than would have been ideal. And I think this pandemic has taught us that we need to plan better, we need to be better organized and better prepared and better coordinated and with better communication, not only within government agencies and between government and industry, but across continents and across cultures, and have a better global response to something like this that affects all of us.
I think those lessons are especially important when it comes to diseases like influenza, which travel very quickly across borders, and then diseases like Ebola, which will probably emerge from a place in Africa, where of course you'll need international mobilization very quickly. So are we applying any of the lessons that we've learned from the SARS-CoV-2 pandemic to these emerging diseases?
I think a lot of the systems have been built on the fly, as I mentioned, that and I think we're still trying to survive the current pandemic. And I don't think we're yet really to the place of looking forward to what could happen next. And I really hope that there can be a sustained political will to do that and to really take the lessons from this pandemic and be much better prepared, not only scientifically, but logistically and in every other way, communication-wise and coordination-wise, to respond better to the next pandemic. Because I think everybody's witnessed how, especially for viral diseases, especially for RNA viral diseases, that the virus is going to evolve. And it's going to evolve not only to improve its transmission and infectiousness, but it's going to evolve to evade immune responses. And so you're going to get variants, as we've seen during this outbreak. And so we need to do better with community education so that people are willing to take the vaccines that are effective and safe without so much hesitancy. And we need to be better at deploying the vaccine around the world, because if we can't immunize the whole world in 6 months instead of 6 years, we will have the same issue coming back around again, because the variants come from ongoing infection. So if ongoing infection is happening anywhere in the world, it's going to continue to raise variants that will make our treatments less effective.
And on the subject of community education, what do you think the biggest lesson that we've learned from this pandemic about what are the best options for community education, what works best, what convinces people to take the vaccine?
The thing that we need the most is trust, and trust between individuals and large institutions, trust between people and their governments, trust between people and the biomedical enterprise cannot be done quickly. So we have to do trust building and education when it's not in the middle of a crisis. And so this has to be something that is going on, forever, really. And the other thing I think we need is just better general population education on biology. I think the understanding of biology in general, which has had such profound advances over these last few decades, is not understood very well by the general population. And so we need to do a better job in our schools and even at the elementary grade level to make sure people understand biology better so they can make their own decisions in a better way.
As a biologist, I support that. So
yes, and it's fun, and I think we have an opportunity here because of what's happened. You know, all these children who've been deprived of a normal education over the last couple of years, they're all sitting at home wondering how when they grow up, they're going to be a viral immunologist and solve all the virology problems going forward, or an immunologist. And so we have a whole cohort of young people now primed, ready to become viral immunologists and vaccinologists, and we need to get out of their way and let them be prepared for what they could do in the future. And I just know there's thousands, if not tens of thousands of young people out there who could really make a big impact 10 or 15 years from now.
Why are you excited to attend this meeting? And what are you looking forward to hearing about?
Well, prior to the pandemic, the vaccinology world or viral immunology world was relatively small, only a few thousand people. Now I think half of the world is a virology expert or has some at least rudimentary knowledge of vaccinology and how immunology works. But there's a relatively smaller group of people who've been devoted to vaccinology issues for many years, and I'm looking forward to seeing them face to face again. I haven't met with my friends and colleagues face to face for a very long time. It's all been over Zoom. So that to me will be the highlight of the meeting, is getting to see friends and colleagues. I think that, I think having gone through this pandemic, everyone going through it together and having so much effort by so many thousands of people applied to just dealing with this virus over the last couple of years has generated information that otherwise would have taken decades and decades to generate. So virtually every conceivable vaccine modality has been applied to this. We've gotten a lot of new adjuvants into the clinic because of this that hadn't been in the clinic before. And so, as opposed to the gradual grind of things through small animal models and large animal models, we're going to have a lot of human data that will help us advance and understand vaccines in a fundamentally different way than we were ever able to before, because having the human data and having several large efficacy trials of data that with different vaccine modalities expressing pretty much the same antigen is going to just be a treasure trove of data and understanding, that will help us understand how vaccines work well into the future. And so seeing some of that data on display and being picked apart, I think is really going to be the beginning over this next couple of decades. I think people are going to go back to this data over and over to try to understand some of the nuances of how the vaccines work.
Yeah, that makes a lot of sense. So you'll be interacting with early career scientists, and a lot of early career scientists will be watching this, and we'd like to know if you have any advice for them.
Science is a career that you can't really entirely plan or predict, and I think the best advice is that you just have to keep focusing in on the biology and asking better and better questions with better and better technology, and to allow yourself the time to make enough mistakes so that you can get to that advance that's just around the corner. And because most of science is a series of failures leading up to a breakthrough, and unless you're willing to live through those failures and persist through the failures and keep going and even try to enjoy some of the failures, because it should have taught you something. That to have a career in science is really a long-term proposition. It's not something that's going to just burst forth right at the very beginning. So you need to take a long view, a long horizon view of this. And people like me, as I did, I recently retired from NIH. I recently retired and left the Vaccine Research Center because there are a lot of very talented 40-year-olds and even young 50-year-olds who have a lot of good ideas, and I wanted to get out of their way so they could start making the decisions and controlling the money and how it was spent and taking this into the next era.
Great. Okay, I think the last question that I have is there anything else you'd like to share with the audience?
I think there's a couple of things that I might want to mention, and that is this issue of young people, not only letting the elementary and middle school and high school students have a place to go to get information and to have doors open for them, but the young scientists in our lab who need a place to go, who need mentoring, I just hope that people can be as open to that as possible in ways that not only include mentoring and advocacy or just education, but also sponsorship, because a lot of people won't get to the next level unless they have doors open for them. It's just not going to be something they can jump into without having access. And so I just encourage people to create that access for the young people coming up.
Thank you so much. I really enjoyed talking to you. I think I'm very excited for this meeting, and it was nice to get the perspective of one of our keynote speakers. So.
All right. Well, I look forward to the meeting too. So thank you.